So now that we've gone over the history of DNA and how we figured out exactly what it was, let's delve a little bit into actually the molecular biology behind it and how it replicates. Okay, so just to reiterate, right, we've got the phosphate group of one nucleotide is attached to the deoxyribose sugar of the next nucleotide, and this results in what we refer to as the sugar phosphate backbone. Okay. Watson, Crick, and Franklin determined that the chemical side groups would form hydrogen bonds connecting the two strands. Adenine forms 2 bonds with thymine, and guanine forms 3 bonds with cytosine. And this explained Chargaff's rules, which is that the amount of adenine is equivalent to the amount of thymine, and the amount of guanine is equivalent to the amount of cytosine. So the structure of DNA is related to 2 primary functions. Number 1, it copies itself exactly for new cells when it's created, and it stores and uses the information to direct all the cell's activities. So when looking at DNA replication, there, again, we sort of had to work our way through models. Okay, the first was the conservation or the conservative model, which is that I have this parent strand of DNA here, okay? That remains intact while a new strand is created. 2 new strands are created and bound to each other, whereas the original strands stay together, and this continues. I have the semi-conservative model, which is that basically I take one strand and I make a copy of it. I take the other strand and I make the copy of it. By the way, by making a copy, I apologize. These run anti-parallel to one another. Okay, so it's its complement strand. Okay, it's not an exact copy, it's the complementary strand of it. And then I have the dispersive model, which is that you have little bits and pieces that are the original versus the new. Okay, so Mendelssohn and Stahl did an experiment, and this supported the semi-conservative model, which was proposed by Watson and Crick over the other 2 models. Basically what they did, sorry, I'm going to get rid of my laser pointer here because it's bothering me and it's being difficult to say the least. Okay, here. So in their experiments, they labeled nucleotides of the old strand with a heavy isotope of nitrogen, N15. Any new strands were indicated with a lighter isotope of N14. The replicated strands could be separated by density in a centrifuge. Each model, the semi-conservative, conservative, and dispersive, had specific predictions on the density of the replicated DNA strands. There's a little video there if you want to watch it. So the first replication in the N14 produced a band of hybrid N15, N14 DNA, eliminating the conservative model. Okay, so we know, otherwise we should have had 2 bands. Okay, the second produced both light and hybrid DNA, eliminating the dispersive model, because the dispersive model would have said we would have had multiple strands. Okay, or I'm sorry, the dispersive model says we should have 1 strand that is because it's everything is a mixture of it. Okay, and that's not what we got. The conservative model says that we should have only light and only heavy, whereas the semi-conservative says that we should have light and a combination thereof. Okay, so this is after the second replication. Okay, so the first replication, we should only have 1 because it's a mixture. Conservative, we should have 2, didn't show up that way. Dispersive, we should only have 1, which it shows up here, but then we end up having we have variations down here. We don't end up with the light only 1, which we do get out of this. Okay, so replication begins at a site referred to as, wait for it, the origin of replication. Prokaryotes, okay, bacteria only have 1 origin of replication, whereas eukaryotes have literally hundreds of thousands of origins of replication. Let's face it, we got to have them. We've got 46 chromosomes, okay, 3.5 billion base pairs. It would happen way, way, way too slowly if we only had 1 starting point. This picture right here, you're going to be drawing me this picture right here. So you might want to pay attention to this picture right here. Hint, hint, wink, wink, nudge, nudge. This is an even better one. So helicase attaches to the origin of replication, unzips DNA. Our bad joke, as always, is how is helicase like a teenage boy? He always wants to unzip your genes. Bum. Okay. and it breaks the hydrogen bonds. Now, this is where we get into why it's really important that there is hydrogen bonding in between the nucleotides, because we regularly break those bonds. Hydrogen is the weakest of the bonds. So we want to make sure that it's something that we can have access to easily. These phosphodiester bonds on the outside that are between my phosphate groups and the sugars are very strong. We don't want that falling apart. We only want to unzip it. It's just, I mean, I know it's a bad joke, but right, like jeans, really strong stitches along the zipper, but then you have the zipper, which makes it easier for you to put your pants on and off. Primers are short nucleotide fragments of DNA, which DNA polymerase will add to nucleotides according to the base pair rules. Primase is the enzyme that creates a primer that can initiate the synthesis of a new strand of DNA, okay? So we go in and we start adding. Primase, an RNA polymerase, adds RNA primer to the strand. Okay, it always goes in a 5' to 3' direction. So I have my RNase primer, and then my DNA polymerase comes on and binds to that and goes along. DNA polymerases are enzymes that catalyze the elongation of DNA at the replication fork. One by one, the nucleotides are added by the polymerase to the growing protein chain, and they just look, they're like, oh, here's a T, I'll add a C. Or I'm sorry, here's a T, I'll add an A. Here's a G, I'll add a C. That's all that it does. DNA polymerase can only attach nucleotides at the 3' end of the growing daughter strand. Replication always proceeds in a 5' to 3' direction because of that. Okay, we can see the label here, 5' to 3', so we are adding it along here. The strands in the double helix are anti-parallel, okay? They run in opposite directions. So each DNA strand has a 3' end with a free hydroxyl group attached to a deoxyribose, and a 5' end with a free phosphate group attached to a deoxyribose. 5' to 3' of one strand runs counter to the 3' to 5' of the other, which you can see here, right? 5 to 3, and this one starts 3 to 5 if we're looking at it in that direction. The 5' always has the phosphate with it. The 3' always has the hydroxyl with it. I remember this because honestly, I know it's stupid, but the hydroxyl has the H, both O and H come before P, so 3 comes before 5. That's how I remember that. It's dumb, but it works for me. So if one strand is known, the other strand can be determined. So I have an A, I go to T, C, G, I'm going to have a C, C, A, T, A, G, G. Okay, we can figure it out. Another DNA polymerase removes the RNA primer and replaces it with DNA. Okay, so DNA polymerase has to attach to a hydroxyl group. It can't start from nothing, whereas RNA polymerase can start from nothing. Okay, so this then goes in and it removes the RNAs and puts DNA in. Then I've got ligase. This forms covalent bonds between the DNA fragments. Okay, right? So I went here and I have new DNA that was put in, right? I have to connect this last one with the new one, and ligase does that. So because I can only add nucleotides to the free 3' end of the growing DNA strand, right? I have an issue. I have my replication fork. One parental strand is 3 to 5, the other is 5 to 3. At the replication fork, the parental strand that is 3 to 5, which means now I'm adding 5' to 3', is the leading strand because polymerases can be used as a template and it just goes. It doesn't have to worry about anything. The lagging strand, which is the 5 to 3' end of the fork, is copied in short segments called Okazaki fragments, right? So this, right, started here and it'll just keep going. It started here and it just keeps going and going and going. This has to go in this direction. So it waits till some unzips, it goes in and it replicates. It waits till a little more unzips, it goes in and replicates. They're each about 100 to 200 nucleotides long, and then DNA ligase comes in and smooths it all out. Okay, it goes through and it adds those bonds that need to be added. This is just a couple, so here you can see, right, this is my leading strand. Okay, it goes along easily. This is my lagging strand, which is going in this direction, which means I have my Okazaki fragments, my DNA polymerase. Eventually, ligase will come in. Oh, I'm sorry, they do have ligase here. Okay, ligase comes in here and it will make a nice smooth chain. So mistakes happen. Okay, and the initial is an error rate of about 1 per every 10,000 base pairs, but DNA polymerases do have proofreading capabilities. They can only add a nucleotide to the growing strand if the previous nucleotide is correctly paired. If a mistake happens, it will backtrack, remove the incorrect nucleotide, and replace it, which makes our final error rate of about 1 per 1 billion nucleotides. Now, that still means we're making approximately 3.5 errors every time our cells replicate and divide, okay? But in the grand scheme of things, that's not bad. DNA molecules are constantly subjected to potentially harmful chemicals and physical agents, right? We've got reactive chemicals, radioactive emissions, X-rays, ultraviolet lights can all damage nucleotides. DNA bases undergo spontaneous chemical changes under normal cellular conditions. So mismatched nucleotides that are missed by DNA polymerase or mutations that occur after DNA synthesis is completed can often be repaired. So each cell continuously monitors and repairs its genetic material, with over 130 repair enzymes identified in humans, which is really amazing if you think about it. In mismatch repair, special enzymes fix incorrectly paired nucleotides. Okay, so for example, there's a defect in one of these enzymes that's associated with a form of colon cancer. Nucleotide excision repair, nuclease cuts out a segment of damaged DNA, and then DNA polymerase and ligase come in and fill the gap. This is used by skin cells when repairing genetic damage caused by ultraviolet radiation from sunlight. And tanning beds, don't use them. So repair enzymes is clear in the disorder xeroderma pigmentosa. They are hypersensitive. These people are hypersensitive to sunlight. Ultraviolet light can produce thymine dimers. Individuals with this disorder, the mutations in their skins are left uncorrected and they are riddled with skin cancer, unfortunately. But when we look at disorders like that, we begin to see the importance of these particular proteins.